Click the bell icon and subscribe to our channel so that you will not miss any classes. Hello, welcome to class. We have studied there are two core processes in biotechnology. The first one is recombinant DNA technology, and upon recombinant DNA technology we will get a recombinant cell, and from that recombinant cell, in order to get a desired product, the second core process is to maintain the sterile environment to get the desired product. And now we shall discuss one of them in detail, that is recombinant DNA technology. And let us understand first how the concept of recombinant DNA technology was developed. And under the heading conceptual development, that means any concepts are not developed as such. By observing something, new concepts are developed. Like way, the recombinant DNA technology was also developed by observing some things which are happening in the natural science. And before going to that, let us understand the definition for recombinant DNA technology. Recombinant DNA technology, it means it is the addition, removal, replacement or repair of genetic material that is commonly DNA. That means in all majority of the organisms, the genetic material will be DNA except some RNA viruses. Genetic material to get a new phenotype. Phenotype means new observable character. The new character will be obtained upon addition or removal or replacement or by the repair of genetic material. And the concept of this, it is possible to replace or repair or addition or replacement of a genetic material. And this happens naturally. Yes, of course, this process, addition or replacement or change in the genetic material, happens naturally upon reproduction. And we know that there are two types of reproduction. One is asexual reproduction and sexual reproduction. Asexual reproduction will not give chance for this. That means the characters or the genetic material is conserved in asexual reproduction. That is from the first chapter, that is reproduction in organisms. We know that there is no change in the characters in asexual reproduction. Genetic material will not change, will not undergo change, because there only the somatic cells are involved in the reproduction. Hence, there is no change in the genetic material. So, there is no chance for this in asexual reproduction. Whereas, in sexual reproduction, the sexual reproduction will give a chance for variations, because in sexual reproduction there is a process called crossing over, where there will be a formation of new characters. In crossing over, what happens? There is an exchange of genetic material happening between the maternal and paternal chromosomes. If if it is maternal and paternal chromosome, they exchange their part. If the genetic material is exchanging, then there will be a new combination. Whenever it is changing, there may be mutation happening, there may be replacement happening, or there may be repair happening. All this may happen in sexual reproduction. But one of the drawback here is it is time consuming. No doubt, it is time consuming. Which means in order to include this, in order to develop some new variants or new organism or a new character, one should observe for more generations. Scientist has to observe it for more generation because the organism has to produce gamete, gamete has to undergo fertilization. After that, the organism should come. From that organism, character should be studied. It is a time consuming process, and also this natural process may include harmful genes sometimes. Sometimes harmful genes may be expressed. Then we will not get a desired product. So understand the topic. So here, in sexual reproduction, there will be a formation of new combinations of the gene or the new characters, which is happening naturally. But here, it is a consuming process to include under the experimentation, and in the natural process, there may be a inclusion of the harmful genes. So, scientists have thought that we can create these variations in artificial or in vitro. In vitro condition, it is possible to overcome this problem by conducting in 3 steps, by constructing the recombinant cell, then introducing or transferring the gene, or we can say it as gene transfer, and gene cloning. We will be studying these in detail further. By these 3 steps, it is possible to create a cell which is possible to express a desirable character or the desirable product, which is a drawback in natural sexual reproduction. So, from there, the concept of recombinant DNA technology, that is, it is possible to create a recombinant cell. Whenever we create a recombinant cell, then only the desirable genes or the gene of interest can be introduced or can be expressed here. That is in the laboratory condition in order to get the product of our interest. Now, the question will arise, how it is possible to do this? That is, how it is possible to construct a recombinant cell. In nature, whenever a aligned piece of DNA, or we can refer it as a foreign DNA. When an aligned piece of DNA introduced into a cell, that means aligned piece of DNA means this is not belonging to the cell. Let's say, for example, I am taking the example of Escherichia coli bacteria. Escherichia coli bacteria is there and some foreign gene, that means this gene is not belonging to Escherichia coli. Whenever it is introduced, an aligned piece of DNA, whenever it is introduced into the cell of an organism, what may likely to be happen? What may happen naturally is, naturally this aligned piece of DNA may not be able to multiply, which means replicate, multiply in the host, as such. If it is present as such, then this aligned piece of DNA may not undergo replication. That means it may not produce its copies. But the DNA of an organism, already there is a chromosome present in this organism. Every living organism will have genetic material, right? So here the genetic material of Escherichia coli will divide, right? So this aligned piece of DNA can divide. Most likely to be, it may not undergo division if it is present as such, not division, multiplication or replication. If it is present as such, whenever it is whenever present in the cytoplasm as such, it will not divide. There is a possibility of replication when it is integrated, when aligned piece of DNA integrated with host chromosome. Whenever it integrates with host chromosome, it may divide. Remember, these are the things which were naturally observed by the scientist in order to develop the recombinant DNA technology. So I am explaining what happens naturally. So here, when a aligned piece of DNA enters into the cell and present as such, it may not divide or it may not replicate. There is a possibility of undergoing replication whenever it is integrated with the chromosome of an organism. This is a chromosome of an organism. Whenever it is integrated with this, that is, integration means if it become a part of this chromosome. If it become a part of this chromosome like this, so here it has become a part of its chromosome, then along with the chromosome, we'll just write it in a block. So along with the chromosome, this is the aligned DNA and this is chromosomal DNA. So along with the chromosomal DNA, this aligned piece of DNA will also replicate. Replicating means it can produce the product also, whatever the gene it is, information it is having, that product will also be obtained. So here it will undergo replication and produces the multiple copies. How much number of this cell produced, that amount of or that number of this gene will also be produced in the host cell. So in the host cell, in the further generation, it will be transferred and it will undergo replication and produces the product. Now you may ask the question, why only with chromosome the aligned piece of DNA can replicate? Why? Because in each and every living organism's chromosome or in the genetic material, in order to undergo replication, you might have studied in molecular biology, during the replication, to start the replication, there are specific sites. They are called origins of replication. Origin of replication. So it is generally called ori site. Ori site. So the chromosome will have the ori site, but this aligned piece of DNA is not having ori site. That's why it is not undergoing replication. Whenever it is integrating with the chromosome of an host, the host chromosome is having the ori site. Whenever it is replicating, along with that, it also replicates the aligned piece of DNA. Not only that, it will be transferred to the next generation. It will be maintained as such. And this process is called cloning. This process is called cloning. Cloning means it is production of, I'll write the definition here, production of multiple copies of aligned DNA. It happens upon replication. Aligned piece of DNA in host and transfer to next generation. Next generation is called cloning. So what I explained here, gene cloning. So aligned piece of DNA, when it is introduced into the host, along with the host chromosome, the foreign DNA will also undergo replication and it will be transferred to the next generation also. The process is called cloning. This will happen in some bacteria naturally. So this was observed by scientists and they make a trial to construct a cell artificially, that is in an in vitro condition, laboratory condition. And from there, the concept of recombinant DNA technology was successful. So from there, there was a construction of recombinant cell, and from there then there was an acceleration given for constructing various types of recombinant cells in order to get various products. So now let us understand how the first recombinant cell was constructed. So here it is just an observation by the scientist, and later on by applying this they constructed a recombinant cell for the first time. Let us understand now what is that cell and how they constructed it.